Okay, thank you, Professor
You're
Pierre.
welcome.
Inventiva is currently assessing the therapeutic effect of IVA 337 on skin and liver fibrosis conditions. Could you briefly explain why the company opted for these two particular conditions?
Yeah, sure. So I would like to come back a little bit to the mechanism of action of IVA 337. IVA 337 is an agonist to the three PPAR isoforms, and PPARs are actually nuclear receptors that control the process of fibrogenesis. So actually this process is somewhere redundant in many organs. You find it in the skin, also in the lung, the kidney, or the liver. And we have actually positioned our compound in two indications where there is a huge medical need. The first one is systemic sclerosis, of course, and the second one is NASH. And NASH is a disease where liver fibrosis makes patients at risk of developing cirrhosis and liver cancer. So this is why it's important also to address this indication.
In previous phase 1 and phase 2A studies, IVA 337 showed a good safety profile. Have there been any relevant adverse side effects associated with this therapy to date?
So very interestingly, this compound has demonstrated excellent safety, both in animal toxicology studies on the long term, but also during our multiple phase 1 and phase 2A clinical studies. So we are confident that we can demonstrate efficacy in patients with very acceptable side effects.
Mm-hmm. Okay. So Dr. Yannick, Inventiva is currently conducting a phase 2B clinical trial, FAST, to assess IVA 337 in Scleroderma patients. Its primary outcome is the measurement of skin thickness, as the drug is expected to improve skin fibrosis in this patient population. Will the potential effects of IVA 337 in other parameters also be analyzed, given that Scleroderma can be a multi-organ disease that affects not only the skin, but also the lungs, heart, gastrointestinal tract and kidneys?
So yes, you're absolutely right. Systemic sclerosis is a heterogeneous disorder which is characterized by the propensity of fibrosis to develop in various organs, but it's mainly targeting the skin. And of course, for a clinical trial and the proof of concept study, we had to work on a homogeneous group of patients. And because the preclinical data were mainly obtained in skin fibrosis animal models, the proof of concept study is focusing on skin involvement, but during the trial, of course, all the other potential organ involvements will be recorded, like, for example, pulmonary functional evaluation to evaluate whether lung fibrosis may develop or change during the trial. So the primary outcome will be skin fibrosis evaluated by the Rodnan skin score and some biomarkers, but secondary outcomes will include the evaluation of all the potential organ involvements.
Mm-hmm. The completion of clinical trials can be hindered by lack of participants, especially on studies in rare diseases. For your FAST trial, you plan to recruit up to 132 participants with Scleroderma. Would you like to leave a message to potential participants on the relevance of this trial and the importance of participating?
Yeah, this is of course a key issue, and we have to recruit enough patients to be able to demonstrate the efficacy of the drug and to demonstrate the risk-benefit ratio for the patient. I think that one of the key points is that the compound is really original regarding its mechanism of action. During systemic sclerosis this last year, many immunosuppressant agents have been developed, and many did fail to demonstrate any efficacy. And for this specific compound, the mechanism of action is really original. Second, the safety profile is very good, and this is something very important for a disease like systemic sclerosis. And I would say finally that we've intensively worked with the company and with many experts to make a very recruitable trial. For example, we are allowing the use of classical immunosuppressant, and EVA 337 will be used on top of immunosuppressant, which is facilitating the recruitment of the patient for such a trial. But of course, we have to mobilize the medical community and the patient, because it's very important to have a clear answer, and to do so, we need to have the right sample size.
Okay, thank you very much.